Hello, my name is Calderón Bracho, and today I'll be talking about Drosophila in Fas2, advancing kidney disease research. But first, let me tell you a little about myself. I'm originally from Venezuela and currently living in Seattle. I'm a rising senior at Rayhan Aviation High School, and I'm involved in 8 clubs such as the National Honor Society, Women Aviation, Environmental Club, the New Technology Student Association, Speech and Debate, LSU, BSU, and Key Club. I'll be president for 2 clubs this coming school year, which are Women Aviation, Women in STEM, and Latino Student Union. I'm passionate about helping others in my community, including teaching young girls how to code, helping young students get excited about science and math, and I tutor over 10 students in my school in subjects such as Spanish and physics. This summer, as a GHLC member, University of Washington intern in the Department of Biochemistry for the 2nd time, helping Seattle Children's Hospital, and currently doing academic mentorship programs to help POC students. I just hope to make change within the environment that I surround myself in and bring awareness in the field of global health. I also hope to keep learning new lab approaches, dive deeper into bioengineering processes, and enhance my STEM knowledge. So what if I told you that flights and humans are more similar than you think? Good evening everyone, my name is Calderón Bracho, and today I'll discuss an intriguing topic in the field of developmental biology, Fas2 and its relevance to development, and specifically its impact on the Malpighian tubules in Drosophila embryos. The understanding of this process not only provides light on the intriguing mechanisms underlying embryonic development, but also provides valuable insights into human biology. Before I delve into the fascinating world of Fas2, I'll provide information on where I conducted my research and its importance. I had the opportunity to work in the Vasquez Lab at University of Washington, Seattle campus, which helps to conducting research on a topic that affects my family and several people in my community, but also all around the world. With that said, the Vasquez Lab is known for being curious about how cells work together. This means the molecular rules that cells utilize to build and shape. Because it is difficult to study organ development in humans, and we believe in many of the rules that cells use to build organs are conserved. We use the model system of Drosophila melanogaster, also known as the fruit fly. Now, let's explore the fascinating world of Fas2 and its significance. Firstly, what is Fas2? Fas2, or fasciclin 2, is a self-adhesion molecule that plays a crucial role in neuronal development and has some guidance. It is found in a variety of organisms, including humans, which have a version homologue of Fas2 in Drosophila embryos. Fas2 is expressed in the nervous system, muscle cells, and the Malpighian tubules. You might be wondering, why are the Malpighian tubules important? Well, the Malpighian tubules are excretory organs and enzymes responsible for removing waste products from the body. They also play a crucial role in osmoregulation, maintaining the balance of water and ions. Understanding the development of these tubules is vital for comprehending the overall physiology of insects and potentially even providing insights into human kidney development. Now, there were several methods used to lead me here. For starters, assemble the plates by putting yeast onto the serve as food for the flies and changing them on a daily basis. For my research, I also did other techniques such as immunofluorescence, which is a powerful technique used in biology to visualize specific molecules within cells. It involves the use of antibodies, which are proteins that can recognize and bind to specific target molecules with high affinity. The process typically includes 2 steps: primary antibody binding and secondary antibody binding. In the first step, a primary antibody is introduced to a sample containing cells. This primary antibody is designed to specifically recognize and bind to the target molecule of interest. In this case, it was Fas2. Once found to its target, the primary antibody forms as an antibody-antigen complex. In the next step, a secondary antibody is added. The secondary antibody is labeled with a fluorescent marker, such as a fluorescein intestate, more known as FITC, or rhodamine. The secondary antibody is produced to specifically recognize and bind to the primary antibody. So, as a result, the fluorescent marker attached to the secondary antibody is brought into the close proximity to the target molecule, in this case, Fas2. When the sample is examined under the fluorescent microscope, the fluorescent marker emits light when excited by a specific wavelength. If a cell expresses Fas2, the primary antibody will bind to it, and subsequently, the secondary antibody labeled with the fluorescent marker will bind to the primary antibody anti-aging complex. As a result, the cell will emit fluorescence when exposed to the appropriate excitation wavelength. This fluorescence allows us to visualize the presence and distribution of Fas2 within the cell. By examining the fluorescent signal, we can gain insights into localization, abundance, and interactions of the target molecule, and further understand the cellular processes in which it's involved. So, how does Fas2 affect the Malpighian tubules in Drosophila embryos? Recent research has revealed that Fas2 is involved in the proper formation and branching of these tubules during embryonic development. It acts as a molecular guide directing the growth of cells within the tubules and ensuring their correct connection and organization. And what I'm trying to find within my research and my thoughts and hypothesis is that I noticed that Fas2 is relevant protein specifically in the neural system of the fly's embryos. But I wonder, what will happen if we didn't have it? We knew that if it's something important in the system, without it, it wouldn't be the same, but how would the embryos react? Will we see the flies with no Fas2 to prove this? So my hypothesis was that if the disruptive rats in the Malpighian tubules exhibit abnormal morphology and impaired function. Importance. So this highlights the critical role Fas2 plays in the development of these excretory structures. By unraveling the molecular mechanisms by which Fas2 influences tubule development, I hope to gain from this deeper understanding of how complex organs form and function within things similar to Fas2. So, furthermore, the findings regarding Fas2 in the Malpighian tubules in Drosophila embryos hold significance beyond the realm of insect biology. Evolutionarily conserved and genes and developmental pathways often have counterparts in humans. Therefore, studying the role of Fas2 in Drosophila embryos might provide valuable insights into the development of function of human excretory organs, such as the kidneys. In conclusion, Fas2, a cell adhesion molecule, plays a crucial role in the development of the Malpighian tubules in Drosophila embryos. By guiding the growth and organization of cells within these tubules, Fas2 ensures their proper formation and function. This means that understanding the molecular mechanisms behind this process not only enhances our knowledge for embryonic development in insects, but also has the potential to provide insights into human biology, particularly the development of excretory organs. As for my acknowledgments, I would like to thank Claudia Vasquez, my PI, Michelle Chicas, Vesta McGannon, and the University of Washington, Seattle campus for giving me the opportunity to dive deeper into something that is so relevant to me and that can make so much difference worldwide. As for my reference, I have my lab notebook, which has different scientific papers that I use to conduct my research and understand, and more deeper understanding within the processes that I use. So if you're interested in taking a deeper look into my research, feel free to do so. Thank you!